Electron micrographs reveal influenza type A to be enveloped viruses, covered with approximately 80% of these route-shaped glycoprotein spikes on hemagglutinin antigen and 20% on mushroom-shaped neuraminidase antigen. The H is a transmembrane protein, whereas N is an integral membrane protein. The inside of the particle is lined by a matrix 1 protein. In addition to the N and H proteins found on the surface of the virus, there are also small amounts of M2 ion channel integral membrane protein present in influenza A virus particles. M2 is not present in influenza B virions. M2 channel protein is inserted into the lipid bilayer membrane that surrounds the viral genetic material. Directly below the lipid membrane of the virus particle is a layer of viral matrix protein termed M1. It is the most abundant virion protein. Inside the virus are 8 segments of single-stranded RNA of negative polarity ranging from 890 to 2341 nucleotides in length. Genetic variation, antigenic drift. RNA viruses are more prone to mutation than DNA viruses. All polymerases, whether they use DNA or RNA as a template, introduce errors as they incorporate an incorrect nucleotide. But DNA-dependent DNA polymerases have proofreading ability. They can remove an incorrectly placed nucleotide and replace it with the correct one. RDRPs, on the other hand, do not have proofreading ability. This raises the overall error rate of the enzyme from 1 error per 109 bases for a DNA polymerase to greater than 1 error per 105 bases for an RDRP, which results in lower enzyme fidelity. RNA viruses have some of the highest mutation rates of all biological entities. Mutations generated by RDRPs may result in mutated viral proteins and subsequently slightly different strains of the virus that may survive better under environmental pressures. Antigenic drift is the gradual accumulation of new epitopes on the H protein and, to a lesser degree, the N protein. Antigenic shift. Antigenic shift does not occur with influenza B viruses because influenza A appears to infect humans and animals, whereas influenza B only infects humans. This phenomenon usually occurs when a pig lung cell is infected with 2 different influenza A viruses, one of non-human origin, usually an aquatic bird, and the other a human strain. Normally, influenza A viruses from birds grow poorly in humans and vice versa. Reassortments between human and bird influenza viruses, however, can produce a new strain of influenza virus that can infect and replicate well in humans. The virus is able to reassort its genes, swapping parts and whole segments of the virus in co-infected cells. For example, reassortment of influenza A viruses result in a new HN strain plus 256 other possible virus combinations. The pig and duck has a mixing or reassortment vessel. The current approach to vaccination against influenza is to use acute virus vaccine that is reformulated annually on the basis of the prevalent strains of virus. The vaccine is moderately effective, reducing mortality in elderly people and illness in healthy adults. Subunit vaccines do not exclusively have to be created through recombinant protein expression. If the virus is able to be propagated in tissues or cell culture, then chemicals can be used to inactivate the virus, and only certain immunogenic viral antigens are isolated and injected at the vaccine. The most common version of the yearly flu vaccine propagates the virus in embryonated eggs, inactivates the virus, and purifies the hemagglutinin proteins. The yearly flu vaccine contains the HA proteins from at least 3 different influenza strains, the identity of which are selected each year based upon the viruses that are anticipated to circulate. Adaptive immune system. B cells initially function as antigen-presenting cells. They interact with antigen via the antigen-specific Ig receptor, promoting endocytosis of the antigen-antibody complex and leading to antigen degradation and processing. After processing, antigen is presented to the T helper 2 cell by the B cell's class II MHC molecule. The T helper 2 cell is then activated to transcribe and translate genes for cytokines. The T cell cytokines then act on the same B cell to divide and form plasma cells or memory cells. Plasma cells produce antibody. Memory cells quickly convert to plasma cells after a later antigen exposure. Antibodies made during infection with H1N1 vaccine bind to H1N1 proteins and prevent reinfection with H1N1 virus, but do not prevent infection with H2N2 virus.